Hi everyone, it's Dr. LeBlanc with another video capture lecture on translation, reading the genetic code. Our learning goals for this lecture are to learn how nucleic acids encode amino acids, to learn how the structure of an mRNA parallels a polypeptide chain, to understand the formation of a peptide bond, to learn the primary, secondary, tertiary, and quaternary structures of a protein, to understand the functional domains of a tRNA molecule, to understand how a tRNA gets charged, to learn important features of the genetic code, and to understand the Wobble hypothesis. How do we read the genetic code? Well, if we take our central dogma of DNA makes RNA makes a polypeptide or protein, we start out with our DNA that produces an mRNA through the process of transcription. A messenger RNA molecule has a 5' untranslated region and a 3' untranslated region, and in between it contains nucleotides that encode amino acids, or the translated portion. We begin with a start codon that is always AUG. All proteins begin with the start codon AUG. This codon, the triplet codon AUG, encodes the amino acid methionine. The code is read in triplets. Each 3 nucleotides encode 1 amino acid, and there are no gaps in between. So the start codon sets what we call the reading frame for all remaining codons. You need to start with the AUG and then read subsequently the next 3 nucleotides, the next 3 nucleotides, the next 3 nucleotides without skipping or inserting any nucleotides in between. Inside the ribosome, peptide bonds are formed between the amino acids. As a tRNA molecule that's charged with an amino acid brings that to the ribosome, it is added in order to the previous growing chain of the polypeptide. Each amino acid contains a central carbon, an R group that varies from amino acid to amino acid, an amino group, and a carboxy group. Here is the next amino acid to be added to this polypeptide. You add it at the carboxy end. This is a condensation reaction. So the formation of a peptide bond releases a molecule of water. And here's the process right here. We remove hydrogens and oxygens and create a molecule of water. And we've formed a peptide bond between this carbon of the carboxyl group and the nitrogen of the amino end of the amino acid. The amino terminal end of a polypeptide corresponds to the 5' end of the messenger RNA, and concomitantly, the carboxyl end of the polypeptide corresponds to the 3' end of the messenger RNA. Note that this diagram indicates that there's a space in between these codons. There is no space in between the codons. There are no nucleotides in between here, no space. All of these nucleotides are right up against each other. So the amino terminal of a polypeptide is the first amino acid laid down, and the carboxy terminal is the last amino acid to be added to the growing chain. This polypeptide chain has 129 amino acids long. How many nucleotides does that represent? How many nucleotides do you need to encode one amino acid? 3. So 129 amino acids times 3 nucleotides per amino acid equals 387 nucleotides. The polypeptide chain is the primary structure of a protein, but this can fold up into things like an alpha helix, like the alpha helix of the helix-turn helix motif that is common in many DNA binding proteins. The proteins can further fold up into a glob-like structure, which is often called a tertiary structure. It can contain motifs of beta sheets and alpha helices. When one or more polypeptide chains associate, such as in the hemoglobin molecule, We call that the quaternary structure of the protein. There are 20 amino acids, each with a different side chain or R group. The smallest is glycine. Its R group is just hydrogen. The largest is tryptophan. It has a very large ring structure as its R group. Amino acids can be divided into different subgroups based on their hydrophobicity and polarity and charge. So these are all the hydrophobic nonpolar amino acids. These are the polar and charged amino acids that are hydrophilic. And then we have some other polar amino acids that are acidic and some that are basic. The tRNA molecule was first proposed by Francis Crick and Marty Hoagland. They proposed an adapter hypothesis. If the central dogma is DNA makes RNA makes protein, then to translate an RNA into a protein, you need some molecule that can go in between, and they proposed that in the 1950s. tRNA is just that adapter. tRNAs recognize the 3-base codon in the messenger RNA by their anticodon, which is down here or here in this diagram. They carry an amino acid that's attached up here that's specific for that codon. Here's a more flattened diagram of a tRNA molecule. That's cloverleaf diagram. The anticodon is down at the bottom here, and the acceptor stem is up here. There is a covalent bond that is formed between the tRNA and the carboxy end of an amino acid. This sequence of CCA at the 3' end of a tRNA molecule is found in all tRNAs. There are also many other modified bases in the tRNAs that we've discussed in the past. How do you charge a tRNA? How do you attach amino acid to a tRNA molecule? The enzymes are called amino-acyl tRNA synthetases. I often refer to them as synthetases. There are 20 different types, one amino-acyl tRNA synthetase for each amino acid. Just one per amino acid. The synthetases catalyze a 2-step reaction involving 3 different molecules: the amino acid, the tRNA, and a molecule of ATP. Here's a diagram of the synthetase with pockets for binding for all 3 of those different molecules. In the first step, an amino acid, the specific amino acid for this specific synthetase, binds, as does a molecule of ATP. The pyrophosphate is released from the ATP, and this ATP monophosphate is attached to the amino acid covalently. AMP is covalently bound to the amino acid, and the pyrophosphate is released. So this is an energy-dependent reaction. The next thing that happened is the specific tRNA binds. Any tRNA that encodes this amino acid will bind. The code is degenerate, and we're going to talk about that in a minute. Many amino acids have more than one tRNA that specifies that amino acid, but all the tRNAs that are specific for an amino acid can bind to the same synthetase enzyme. When the tRNA molecule binds, the AMP is released, and the amino acid is covalently attached to the tRNA, a nice tight bond. After the tRNA is charged, it's released from the synthetase, and the synthetase can go on and charge another molecule of tRNA. There are lots of checks and balances in this process, and if you want to study this enzyme further, it would be very rewarding, because this is a very interesting enzyme. It's by far my favorite enzyme out of all the enzymes. Okay, so the recognition between the tRNA and the mRNA occurs via hydrogen bonding between the nucleotides of the anticodon loop and the codon nucleotides in the messenger RNA. This is the codon for phenylalanine, and the phenylalanine anticodon on the tRNA will bind here and bring this amino acid into the ribosome at the right time in the right place. tRNAs are named according to the amino acid they bear, so this is a tRNA Phe or phenylalanine. This is a tRNA Pro. Notice that the tRNAs are anti-parallel to the messenger RNAs. And notice again that there is no gap here. The Wobble hypothesis. What is the Wobble hypothesis? Francis Crick proposed it in 1966 after he examined the genetic code when it was finally figured out. In the codon, anticodon recognition that we just saw in the last slide, the first two positions pair strictly according to the AUGC rule. However, the third position can actually wobble or move a little bit and thus tolerate certain types of mismatches. So there's a potential in the process of reading the genetic code in the ribosome that mistakes would be made. But if you look at the code, the genetic code, you will notice that all of the codons that dictate a particular amino acid often have the first two nucleotides the same, and the third nucleotide is the one that varies. So anytime you see a UC, it means serine, whether or not it's UCU, UCC, UCA, UCG. All of these amino acids encode, all of these nucleotides encode the amino acid serine. So if you look at this table, you'll notice that the code is universal, almost, and that the code is degenerate. There are synonymous codons, like we just looked at for serine. Phenylalanine has these two. Leucine has these two. Actually, leucine has these 6. Isoleucine has these 3. Methionine has only 1. The AUG is the only codon for methionine. It's also the start codon. Notice also that there are 3 codons that do not encode amino acids. They are the stop codons, UAA, UAG, UGA. So if you were going to memorize any codons from this chart, you should probably memorize the AUG, which is methionine, and maybe also UAA, UAG, and UGA as the stop codons. So here's another depiction of the Wobble hypothesis. This is the Wobble base, the last base in the codon. The Wobble base of the anticodon loop can mismatch. So if it's a G, sometimes it could base pair to a C or a U instead. If it's a G of the anticodon, it could base pair to a C or a U of a codon in a messenger RNA. And if it's a C, it could possibly base pair to a G, an A, etc., etc., etc. Inosine is a modified base. So in summary, the genetic code is read in triplets. 3 nucleotides equal 1 codon, which is translated into 1 amino acid of a polypeptide. There is a single gene, a single start codon, it's AUG, and it encodes the amino acid methionine. The code is read in groups of 3 nucleotides from a messenger RNA without any gaps. This is called the reading frame. Because the code is read in groups of 3, any given sequence of nucleotides will have 3 reading frames. That may take a little while to sink in. An open reading frame means that a given sequence contains no stop codons. Long open reading frames in the genome indicate the presence of exons or even entire genes. There are 3 stop codons, UAA, UAG, and UGA. The anticodon loop of a tRNA contains 3 nucleotides that are complementary to the codon. A tRNA has a stem acceptor arm that ends in a 5' CCA 3'. This is where the amino acids are covalently attached by the group of enzymes known as synthetases. A tRNA with an amino acid attached is called a charged tRNA. Polypeptides have an amino end that corresponds to the 5' end of the messenger RNA and a carboxyl end that corresponds to the 3' end of the messenger RNA. Proteins have 4 levels of structures. The primary structure is the sequence of amino acids that corresponds to the sequence of the nucleotides of a gene. Amino acids have an amino group and a carboxy end, just as nucleotides have a 5' and a 3' end. Amino acid R groups are attached to the central carbon. They have a variety of characteristics based on charge, hydrophobicity, polarity, and size. There are 20 different amino acids, and each amino acid has its own synthetase. However, all amino acids except methionine have more than one tRNA because there are 60 possible codons. This makes the code degenerate. Each synthetase will charge all the tRNAs for a given amino acid. Synthetases are Dr. LeBlanc's favorite enzyme. tRNAs bind to messenger RNAs via complementary base pairing, and the Wobble hypothesis explains why the genetic code has some redundancy, but very rarely in the first or second positions.